afterwards as the chat. Okay, Zareen, why don't you give it one more try? And if it doesn't work, we're going to put you on for later
Okay.
on, okay?
Okay, I'm so sorry. I don't know what's going on.
Yeah, Zoom is a bit finicky sometimes. It's not your fault. We can see your screen. That looks great.
Can you see
And
it?
yep, but can you go to the next slide? Oh, perfect. Okay. So,
It's a little slow, so
yep. Okay, but just go to your first slide and why don't you kick off and then we'll try and stay on time, okay? So thanks very much, Zareen, for
okay.
giving us the first talk today.
I'm just gonna full screen. Give me a minute. It's really slow on Zoom for me. I apologize. Okay. So my protein is calcium calmodulin dependent protein kinase 1G. There's my composal script, and I superimpose the PDB and AlphaFold model together just to give you a glimpse of it. So for function, it's activated by calmodulin, and calmodulin needs calcium. Calmodulin binds to the calmodulin binding domain, and that releases the regulatory domain pseudosubstrate inhibition on the kinase domain. So then CAMKK can phosphorylate the activation loop in the kinase domain. It's a serine threonine kinase. It's expressed highly in the brain, especially in the limbic system, a bit in the ovaries, and a bit in the kidneys as well. Its main job is it goes and forms lipid rafts and dendrites, especially during early development in cortical neurons. And it does this through dendritogenesis via actin polymerization. And it's anchored to the Golgi and PM, as through Phoebus I saw it was non-cytoplasmic. You can see highest in amygdala, cerebellum, a bit in brain, ovary, and a kidney. For structure, a lot to take in, but I want you to pay attention to this and this here. For the kinase domain, I found 4 possible membrane binding regions, and they're relatively close to the 2 of the MET stops and the activation loop PET stop. There is a possible end-term myristylation, but it's non-consensus, so kind of unlikely. And then there are 4 progressive polymethylation sites, and this is like critical for it to get sorted into the lipid rafts. But the most important thing is this S prenylation at this CAX motif near the C terminal. Without it, it doesn't get sorted into the PM or Golgi and dendrites. And you can see the CAX motif has the highest score in GPS lipid as well. For signaling pathway, there's 2 possible ways. It can go for initial outgrowth of neurons and dendrites, about 100 micrometers. It'll do this through like activating PIP, calcium, and getting CAMK1G with RAC, GTP, and STEF, and promoting actin polymerization and leading to dendritic arborization. And another pathway it can happen through is dendritic arborization past 1,000 micrometers. It'll do this through the NMDA receptor pathway, leading to Krebs-dependent phosphorylation and translation of these 2 unique proteins. One of them leads to increased concentration of beta-catenins, leading to actin polymerization branching. And another one is a protein that inhibits the inhibition of RAC GTP, which again, similar to this here. And I just want to show that CAMK1G has similar C terms compared to the RAS protein. Lots to take in here, but I want you to focus on the C term area here. This disolip pred said this area was disordered in lipid binding, which makes sense as that is where a lot of the palmitoylation and the prenylation is located. From MODA, it looks like there's nothing, but the PDB structure has the C term truncated just because it's so disordered. It doesn't multimerize, so I don't think that could really affect lipid binding that way. And then there is the lipidation sites on the motor membrane. For disease-linked mutations, I saw that was observed in the germline of women that developed early onset luminal breast cancer. It was a single nucleotide variation in the codon, and they would code for this serine 57 normally. Which could get hyperphosphorylated in another case as well. You can see here it has a mutation in that very small single nucleotide variation case and amplification in other cases. It's also increased in HER2 cancers and has high polygenic risk score in schizophrenia, and it's related to stress and depression as well, which makes sense if you remember it's located in the limbic system. And I did the Pimal model of the serine 57. I couldn't find it in Pimal, so I would guess it would be right around here. and the serine 177 there. It wasn't there either, so I just did amino acids close to them. So this is what it would look like in space. So in space, they're generally close together in the 3D structure. This is my works cited.
Great.
Perfect.
Any questions,
anyone-
Zareen? Feel free to ask in the chat space. There's one there. Can you see the question?
How do you think P57 affects kinase activity? Let me go back. I think it affects kinase activity since it's so close to the active site. It could possibly inhibit that pseudosubstrate inhibition of the regulatory domain. So then you just wouldn't need comodulin and calcium at all, and MAPK can just activate it whenever it pleases.
Great. Thanks so much, Zareen.
Yeah,
I'm
of course. Thank you.
Unshare your screen and we'll move to Tyler. And there may still be questions in the chat space, Zareen, so feel free to check it out and answer in the chat space while other people talk. Thanks. It looks great, just full screen. You're good to go.
Perfect.
It's just loading. Looks perfect.
Right on. Thanks for everybody being here today. I'm going to talk to you a little bit today about TLK1 kinase, or tussled-like kinase 1. So this is a serine-threonine kinase found in most bodily cells in all multicellular organisms, and it is localized particularly to the nucleus. It has many functions and is involved primarily in DNA replication and has been shown to be most active during the S phase of the cell cycle. Acting as a hub, it interacts with many proteins, particularly when dimerized with itself or its sister protein TLK2. When active, it has been shown to be involved in the phosphorylation of histones, interaction with cell cycle checkpoints, downstream stabilization of microtubule assemblies, DNA repair, as well as telomere extension. As of this presentation, unfortunately, there are no known experimental structures elucidated for this protein, though the alpha-fold structure does exhibit a high degree of confidence in its various domains. Its kinase domain is very similar to that found on TLK2, which does have an experimental structure, and the two kinases share a 96% identity within this domain, with 84% overall conservation between the two. The domains include a nuclear localization sequence on its N-terminal portion of the protein, two coiled domains from amino acids 244 to 277, as well as 403 to 441, as well as that kinase domain from amino acid 456 to 734 with 760 amino acids overall. Regulation, so this thing has been shown to be activated by cis autophosphorylation events in its kinase domain, and phosphorylation events here allow for conformational changes in its coiled domains, allowing for cis and trans phosphorylation, which are shown in green, as well as at its C-terminal tail. Interesting, no exogenous phosphorylation is required for the activation. This autophosphorylation process has been shown to be aided by a protein called LC8, which stabilizes those coiled domains and facilitates dimerization to the active form in the nucleus. Conversely, TLK1 has been shown to be inactivated by another protein called CHK1, which phosphorylates site serine 695 in the kinase domain of the protein in response to DNA damage. Nuclear in origin, TLK1 has oncogenic functions, and its primary substrate, a protein called ASF1A, shown there in orange, is phosphorylated at many sites and gives the protein an overall negative charge, allowing for interactions with histones H3 and H4, promoting replication coupled chromatin assembly. It's also been shown to associate with Aurora B kinase, shown there in that teal blue, to stabilize chromosomes during mitosis. Other interactions include the TLK1 RIF1 complex, promoting telomere extension, mediated interaction with ASF1A with RAD53, which is a critical cell cycle checkpoint, as well as NEK1 phosphorylation at threonine 141, which responds and facilitates DNA damage repair. And then finally, to prevent the previously mediated CHK1 inhibition that was discussed briefly on the last slide, TLK1 acts in unison with the RAD9 complex, in complex with RAD1 and HUS1, to regulate the localization and sequesterization of CHK1 inhibition. Being involved in having potential oncogenic functions, TLK1 has been shown to be rapidly inactivated due to double strand breaks, and this is caused by CHK1, phosphorylating it at serine 695. Mutagenesis studies have showed that mutations at this site from a serine to an alanine impairs this downregulation, allowing for the replication of damaged DNA. In addition to that potential oncogenic function there, meta-analyses have been done of TLK proteins, and they have found that these mutations also provide a broad spectrum contribution to intellectual disabilities, and they identified specifically contributions towards schizophrenia and autism spectrum disorder, which was published in the paper in Nature Neuroscience there. And that's all I have, and I'm happy to take any questions if there are any.
Okay, any questions? There's a question in the chat space. Emmanuel asks, do you know which domains have low confidence in AlphaFold?
So the AlphaFold domains that show low confidence are those of the nuclear localization signals. When I was looking at some of the papers, it's still mostly unknown where exactly these things are. They think it's in 3 different consensus domains, as there are 3 spots along the N-terminal part of the protein that display a high amount of arginine and lysine. Though the rest of the domains, particularly the cold domains and the kinase domain, are have strong confidence as far as AlphaFold is considered.
Okay, great. Just unshare your screen there if you can. And great. And perhaps you can answer the remaining questions in the chat space. That was great. Thanks. Can Thomas present? Oh, great.
Okay. Hello, everyone. Today I'll be presenting on PIP4K2C, which is an isoform of the PIP4K family. And we could see on the bottom left, we have a AlphaFold model of what the protein looks like. Here are some of the basics. PIP4K2C is a monodomain protein, which is just the single peptide and then the whole PIPK kinase. And based on the study, rat study, unfortunately, PIP4K2C is predominantly found in the cytoplasm and endoplasmic reticulum, circa this study. Overall, PIP4K2C is rather ubiquitously distributed within different types of proteins. Strangely, more priority is focused on neurons and kidneys over skeletal muscles. PIP4Ks also typically catalyze the reaction PI5P plus ATP gives you PI45 P2 plus ATP. And it's important to note because this is the same PIP2 used in insulin signaling by, well, first you have DAG signaling with PLC, and you have PI3K for your PIP3. Those scenes aren't always so simple. I wish they were, but it's not. A 2016 study found that PIP4K isoforms metabolize both ATP and GTP at different rates. And in the PIP4K2B isoform, it found that it hydrolyzed GTP at a 5 times faster rate than it did with ATP. But unfortunately, it was unable to get a good scale balance for hydrolysis for the PIP4K2C isoform because there's a minuscule amount of enzymatic activity. On the right, there's a X-ray crystallography structure obtained in 2022 featuring the AMP-PNP, which is a non-hydrolyzable analog of ATP. I'll get to that on the next slide. Now, things are even more complicated. The second study confirmed the finding that PIP4K2C is enzymatically dead. However, it also found that knocking out all 3 PIP4K isoforms resulted in an increase of PI5P concentration, an increase of PI45P2 concentration, and a decrease of PI4P concentration, which seems weird on the surface. How can you have a product and substrate increase in concentration at the same time by knocking it out? Well, what this team did is the mutated residues 69 to 75 in PIP4K2C and reconstituted into a triple knockout cells, and was unable to restore the PI4P and PI45P balance. And however, if you reconstitute the wild type PIP4K2C into the triple knockout cells, you're able to restore the PIP2 and the PI4P concentrations. The reason why is because it's noticed how PIP4Ks actually interact with PIP5Ks, which is responsible for converting PIP4P into PIP2. Now, the only problem is the whole PIP5K family is a dark kinase family, with the only protein crystal structure known is from Daniel, which is the zebrafish, which is unfortunate because we don't know much about the interactions because the structure is not known. Now, here's the interesting bit I found. The green highlights where the 69 to 75 motif is, where the blue highlights where the tip of the alpha helixes end in the first study with the 8 AMP PMP. So it's interesting to me how the catalytic site is so close to where this interaction with the PIP4K and PIP5K occurs. Now, here's the overall structural analysis. Unfortunately, structural prediction revealed no predicted fatty acid anchors. Both MODA and pred predicted lipids, so a lot of the lipid-associated regions on one side of the protein. And with that, 3 PIP stops, 2 acetylation stops, and 2 ubiquitination stops were identified. Although 1 PIP and both acetylation stops were on a single peptide. However, there wasn't necessarily the case given QPRM and phosphosite disagree on some sites, one being a phosphorylation on serine 324. And this is just me doing all the calculations, bunch of boring graphs, and I even combined phospholipid and Phobius because, yeah, it is on a similar scale. Now, with all that data, I finally came to this structure, and you can see the MODA is red here and here. This would be the single peptide. On my screen, PIP5K1A binding is in the back. We'll get to that. Blue is the potential phosphorylation sites back here. We'll get to that. And orange is the potential ubiquitin sites. One there's the single peptide, and one there at the back. Now, if I turn to the side, a lot is revealed. Suddenly, you have this whole plane where it's possible phospholipid binding, and this is a low confidence single peptide, and this is a low confidence loop. So those parts don't matter as much. Back here, we could see this PIP5K1A binding site, which is near this potential ubiquitination site. So possibly, by ubiquitinating PIP4K, you would inhibit its interaction with PIP5K. It's just inhibiting an inhibitor. So my conclusion is one of the main reasons why this protein is so complicated is because there's a lot of uncertainty, especially with PIP5K, because the whole family is an unknown kinase.
I think we better wrap it up there, Thomas. Thanks
Yes,
so
that's
much.
all.
Okay, and maybe you can answer questions in the chat space. Please do ask Thomas questions there, but I think we're out of time. If you could unshare your screen, and then
Yep.
Raj, if you could share yours. Lots of data there. That's great. Thanks so much.
Welcome.
And yeah, do ask questions, please. There was a lot of content there, so feel free to. Okay, that looks great. Just full screen. Okay, it's almost there. And
All right.
great to see you as well. Feel free to show your face if you're giving a talk as well. So that's great. Feel free to start. It's not full screen yet, but we can see the slide pretty well. So you could work with this view if you wish, or if you can click slide show maybe one more time.
One more time.
Well, or maybe you got to switch your screens, but I see only the presenter view. But it's okay. You can scroll through that if you prefer at this stage, maybe.
Oh yeah, I see. I don't know how to like fix that.
Okay, that's fine. You just move from slide to slide using this view. That's fine. And go ahead and start.
Okay. All right, so my protein is microtubule-associated serine-threonine kinase 2, and this will be a brief overview. So the reaction that it catalyzes is going to be just phosphorylating via ATP serine and threonine residues on proteins. And so one of the key functions is that it phosphorylates those serine and threonine residues on microtubule-associated proteins. So for example, utrophin over here is a microtubule-associated protein, and its MAS2 is going to phosphorylate its C-terminal domain. And this is going to alter utrophin affinity for F-actin. And so this is going to regulate your microtubule structure within the cell. An activator of it's also an activator of cell survival, division, and migration. And this is primarily through it being a key inhibitor of lipid phosphatase P10, which is an important tumor suppressor. It's also present in spermatid flagellum maturation. For the expression and localization, I found that it was nearly ubiquitous and found in almost all tissue types. And this indicates that MASP2 has pretty broad functions. It's also localized to the cytoplasm, closer towards the inner plasma membrane leaflet, but it does not bind the phospholipids themselves. It rather binds to transmembrane proteins or peripheral proteins near the plasma membrane.
Can you go to your next slide, please? We still see your first slide.
Oh, I'm so sorry.
Might need to just at the bottom click maybe to the right. Or maybe scroll down. I'm not quite sure.
Just give me 1 second.
And if we do run out of time, I'm going to ask the last speaker whether or not Grace, I guess, wants to present at the end or next class. Okay, we can see slide 4 now. That's great.
Okay, great. So this was just the expression, and I just mentioned that it's pretty ubiquitous. Moving on to the, oh, so did you guys see this slide?
No, we see it now though.
Okay. For the structure, so there's 2 main domains. There's the serine-threonine kinase domain, and there's the PDZ domain over here. So the serine-threonine kinase is an AGC. It's part of the AGC family kinases, and it's at these amino acid positions. It has highly conserved ATP and magnesium binding sites through these, and a key exception to this conservation of the overall AGC kinase is that this polyglycine loop is actually converted to a serine at the first glycine here, which is found in all isoforms. And so phosphorylation of this, which is present in MASP2, will modulate the kinase domain activity, which is this over here. There's also a class 1 PDZ domain at these positions, and it's highly conserved among all isoforms as well as species, primarily because it's a key protein-protein interactor with P10, and the binding motif is this over here. And so this image down here shows us that this green portion is the PDZ domain, while this orange portion is just a C-terminal part of P10. So for pathways and regulation, MASP2 is an inhibitor of P10 because MASP2 phosphorylates the C-terminal domain of P10 and decreases its activity. So this is P10 over here. And so if you inhibit P10, you're going to get a buildup of IP3. And so building up IP3 will activate AKT, and it's going to activate the mTOR pathway. And this overall will lead to cell division and survival. So for MASP3, threonine 389 is phosphorylated by pKa, and this is an extremely major regulatory site for MASP3. And so I wondered whether or not this would be extremely important for MASP2 as well, and if it'd be also present. And so it actually is present. I ran a BLAST search, and it indicated that threonine has just shifted over to position 354 for MASP2. For diseases and mutations, MASP2 is an extremely important protein when considering cancer because the overexpression of MASP2 leads to cell survival, and this is by suppressing apoptosis through AKT. And MASP2 knockouts would result in reduced rates of tumor growth in mice, and so therefore we conclude that MASP2 is an oncogene. Additionally, protocadherin liver, kidney, colon, and MASP2 interactions is a key molecular switch for contact inhibition. And what that means is basically while protocadherin and MASP2 are complexed, it's going to prevent the migration of metastases. And the mechanism is currently unknown, but it's likely due to the microtubule network, which is regulated by MASP2. And this is my composal.
Great. Thanks so much. We're running a little short on time, so I'm going to ask people to ask Raj questions in the chat space. And if Parco can start his presentation next, that would be great. Great. It's coming up. And just feel free to show yourself if you wish.
I don't actually really have a working camera right now, so I can't
Okay,
really
that's
do that,
fine.
but
No pressure, but we can see your slide. Yeah, if you can, full screen,
is this
perfect. Go
good?
for
Is
it.
it full screen? Okay. Hello, my name is Parco, and then this is poly A nuclease 3, a pseudokinase story. So let's start off with an overview. So PAN3 is an 887 amino acid long 95 kilodalton protein. It is a ubiquitous protein that's found in the nucleus and cytoplasm of almost all eukaryotic cells. It exists as a homodimer that forms complexes with many proteins, for example, PAN2, an exonuclease. And it participates in the mRNA deadenylation, in other words, of degradation of the poly A tail of mature mRNAs. As I said before, PAN3 participates in pathways of mRNA degradation, and while PAN3 is catalytically inactive in these pathways, it mostly plays the role of an adapter protein, being recruited to mRNA by poly A binding proteins or TNRCs. It then acts to bring like PAN2, an exonuclease, to these mRNA protein complexes, where PAN2 shortens the poly A tail until it's roughly 50 to 100 nucleotides long. And this is how the PAN3 PAN2 complex works. And it plays a role in general mRNA degradation via interactions with poly binding proteins, but it also has a role in microRNA gene silencing through its interactions with TNRC6s. Here's a little schematic of the complex. As you can see here, there's a dimer of PAN3 attached to a poly A binding protein, and PAN2 over here in red, shortening the poly A tail. And it's important to note, actually, that without PAN3, you don't see catalytic activity of PAN2 in vitro or in vivo. Moving on to the structure, on the left is a PAN3 dimer from the PDB. It's from Drosophila, so it's not perfect for humans. And on the right is the AlphaFold predictive structure for a human PAN3. And I've highlighted important domains, such as here in red, I don't know if you can see that, is the zinc finger domain. And what it does is it helps PAN3 bind to the 3' untranslated region of mRNAs. And we can see that in this figure here. It helps to bind this 3' section of the mRNA. And then next up, we have a PAM2 motif here in blue, and this one helps mediate the interaction between PAN3 and poly A binding proteins. And it also it's flanked here and here by a cluster of phosphorylation sites, and it's a very important regulatory region, which I'll get into later. Then we have in purple the pseudokinase domain. And the pseudokinase domain is actually catalytically inactive due to substitution of many key residues for kinases, right? But it retains the ability to bind nucleotides. So the pseudokinase domain, PAM2, and zinc finger all work together to coordinate PAN3 onto mRNAs and to orient it in the proper way so that it can bring mRNAs to PAN2, which is an exonuclease. And now we have the final two domains I've highlighted. First off, we'll start with the coiled coil in yellow, and this mediates the dimerization of PAN3. And actually, it's important to note that the dimerization of PAN3 isn't required for association with PAN2, but when it does dimerize, it makes a little tryptophan binding pocket here, where TNRC6s, which are previously seen in microRNAs, where they can bind there and be recruiting PAN3 to these complexes. And finally, we have here in pink, the knob domain. And the knob domain here primarily interacts with PAN2. And as you can see here on the structure, the knob domain is actually really close in space with the pseudokinase domain. So the pseudokinase domain would bring the mRNA here right next to the PAN2, which can then degrade it. Moving on to the MODA and the phosphosites, one of the first things you can notice are here the clusters of phosphorylation sites, and they're just flanking PAM2. And these phosphorylations reduce the affinity for PAN3 for poly A binding protein, reducing the amount of PAN3 recruited to mRNA targets and slowing down mRNA turnover. And then on the MODA, there were actually many positives around protein interaction domains or DNA interaction domains. And I'm thinking these could be false positives, as Swiss Palm indicated no lipidations of this protein, and it's cytoplasmic. And so these false positives could be due to hydrophobic interfaces, not only binding to membranes very well, but also mediating protein-protein interactions very well. And additionally, I saw a lot of arginines were flagged too, and while arginines bind to phosphoinositides in the membranes very well, they could also bind very well to the phosphate backbone of DNA. And then here are my references and questions. Dr. Overdoon, you're muted if you're saying anything right now.
Yeah,
Okay.
Sorry about that. That was great talk. Lots of data there. And I apologize that we're running a little short on time. So I'm going to ask you to respond to questions. Keep asking questions in the chat space. That's a great way to save a bit of time here. And is Lauren able to share slides?
Yeah, I'll be right there.
Okay, just unshare your screen there if you can, Parco. Thanks again for a really robust presentation. And then maybe I'll just try and change this for you. Okay, so I'm going to unshare yours. And then Lauren, you can share your screen now.
Perfect.
Okay, and thanks again for asking questions in the chat space. I do want you guys to ask away, and you are evaluated on our participation as well. So feel free to ask at least one question today if you can. And Lauren, nice to see you and nice to see your slides. Go ahead, it's your show.
Okay, perfect. So today I'll be talking to you about my protein, which is MAP3K10 or mitogen activated protein kinase 10. So if I can, sorry, I had to download a PDF because my pictures weren't like uploading. So to begin, MAP3K10 is the first kinase involved in the activation of the JNK pathway. It is a serine threonine tyrosine kinase and belongs to the enzyme class 2.7.11.25. So the JNK pathway is a C-Jun N-terminal kinase pathway. And so in response to cytokines, growth factors, or external stress, MAP3K10 will go on to indirectly activate the transcription factor C-Jun, which will work in conjunction with activator protein 1 to induce neuronal differentiation, axon growth, and apoptosis. So it's mainly expressed in the brain, as you can see in this picture here, and it is subcellular localized to the cytoplasm. For the structure, the primary structure is at the top here, and it highlights the SH3 domain, the kinase domain, and the leucine zipper domains. So the SH3 domain is found from amino acid 16 to 81, kinase domain from 81 to 360, and the leucine zippers are from 384 to 405 and 419 to 440. So the leucine zippers are required for homodimerization and autophosphorylation, and then therefore activation of the kinase. The SH3 domain would likely bind to an SH3 protein that is already bound to the receptor that is receiving the signal in order to propagate it down further along the cascade. There's also an ATP binding pocket at 104 to 112. And so unfortunately, there is no fully crystallized structure of this protein yet. So here in the alpha fold, the places with the largest confidence is just the SH3 domain, which is the only PDB file that there is for this protein. You can see in the Phoebus plot that there is no signal peptide, and there is a very relatively low chance of a transmembrane domain. The MODA and DLPR plot suggest the main membrane-associated region is from 400 to 900. Therefore, we can assume this protein is most likely an intracellular peripheral-associated protein. And my composal is down there. Next, we'll move on to regulation. So my protein is regulated by post-translational modifications like phosphorylations, which are seen at 489, 502, and 830. So the first two phosphorylations I mentioned occur during the dimerization and activation of the kinase. So since there isn't a whole lot of research about this protein, the only other method of regulation I found was at the transcriptional level. So there is DNA methylation that plays a role in the regulation of my protein. It was found that DNA methylation of the gene was less than normal in patients that have intracranial aneurysm. So therefore, we can assume DNA methylation must play a role in regulating proper gene expression of this protein. Finally, we'll move on to the disease-linked mutations. So according to the plot here by CBio, there's a large amount of gene amplification found in uterine carcinosarcoma, which had the highest frequency. The next highest frequency mutation was a missense mutation caused uterine corpus endometrial sarcoma. And then down here on the other plot, there along the same lines was a truncating mutation, which is seen in red, which was proline 504 switched to a leucine, and it was seen in high frequency of uterine and stomach adenocarcinoma. These are my references, and I'll answer your questions in the chat.
Great, Lauren. Thank you so much. And we saved a bit of time there, and that was very clear and careful work. Thank you so much. And I'm going to just ask the next presenter, Lane, to show her screen, and we'll just keep rolling. Keep asking questions in the chat space. That's great. And I'm just going to unshare your screen there for you, and then we can let Lane go ahead. Hi, Lane. Are you there? Ah, perfect. Okay, we can see it. And can we hear you?
Yep.
Okay, great. And it's not full screen, but we
Oh,
can
no?
see it pretty well.
Yeah. How's that?
Perfect.
Great.
Go.
All right. So my protein is STK3. It's also known as serine threonine protein kinase 3, and it's a member of the STE kinase family. And it plays a critical role in the kinase cascade of the hippo signaling pathway, which regulates organ size through the control of apoptosis and cell proliferation. So in response to cellular stress, STIK3 and its homolog STIK4 will dimerize and complex with the regulatory protein SAV, which will then go on to phosphorylate the LATS1 and 2 kinases alongside their regulatory protein MOB, which will lead to the phosphorylation of the oncoprotein YAP, which is then rendered unable to enter the nucleus and promote the genes that promote cell growth and inhibit apoptosis. Some more recent studies have also shown that when caspase 3 is activated during apoptosis, STIK3 is cleaved near the C terminus, losing its regulatory domain, as well as its nuclear export signal. STIK3 will then be able to enter the nucleus and induce chromatin condensation by phosphorylating the histone H2B. So STIK3 is known to reside within the cytosol and the nucleus. Phileas, Phoebus, deep TMHMM, and literature all suggest STIK3 is globular and not associated with the membrane through any transmembrane regions. Literature reports a high expression in placental, skeletal, and liver tissues and more modest expression in the heart, brain, and the lungs. As well, using data from the GTEx project, the dark kinomes kinase expression application reports STIK3 is present at the 50th expression percentile in the uterus, cervix, ovaries, adrenal glands, liver, and skeletal muscle among all kinases. So STIK3 is 491 amino acids long, weighing in at about 53 kilodaltons. You can see here this tightly organized kinase domain by the N terminus. Close in space to the active site will be ATP binding sites, as well as the STIK4 dimerization site, which is located on alpha helix G. As well, there are the two coiled regions, and the one more to the C terminus here will contain the SARA, or the SAV-RAS-F hippodomain, which is where a lot of interactions with regulatory proteins will form. STIK3 was not predicted to be lipidated at any site according to any available predictor program. Here we have the MODA output for the AlphaFold structure with regions predicted of having a high likelihood of membrane association here in red. As you can see, these sites are quite dispersed and they're mostly localized to the SARA domain here. So that suggests that protein binding at the SARA domain occurs through hydrophobic interfaces. Phosphosite yields 19 phosphorylation sites, 16 ubiquitination sites, and just 2 methylation sites. So experimentally, the phosphorylations of threonines 117, 180, and 384 are said to be the most important for regulation. In order to be fully activated, STIK3 needs to undergo a process of trans-autophosphorylation, where that homolog STIK4 phosphorylates its threonine 180, leading to the assembly of the active site. And that dimerization and autophosphorylation process is thought to be facilitated with the help of SAV1. As well, STIK3 activity is enhanced and inhibited by a group of proteins known as RAS-F, or the Ross association domain family. RAS-F1A, F6, and F5 have been shown to block STIK3 autophosphorylation and dimerization through an association with the SARA domain. In contrast, ROS F1A and F2 can stabilize the active dimer and enhance kinase activity. As well, they can protect that threonine 180 from dephosphorylation by PP2A. The kinase ACT, which is involved in cell division, can phosphorylate threonines 117 and threonine 384 on STIK3, preventing dimerization, RAS F1A binding, and allowing the oncoprotein RAP1 to form an inactive complex. As well, methylation of arginines 461 and 467 at the SARA domain can inhibit STIK3 by blocking dimerization. Here are just some references, and if you have any questions, just let me know in chat.
Great, thank you. That was very clear. I really appreciate it. And are there any questions? Maybe we have time for one question if anybody wants to unmute and ask. Otherwise, feel free to ask in the chat space. And maybe we'll ask the next presenter. Kelsey, are you here?
Yeah, I'm here.
Oh, great. Feel free to share your screen.
Okay.
The talks have all been great. You guys have worked really hard. Thank you so much. And I look forward to another full day on Wednesday. And I think Grace is going to go on today as long as we don't go too late. So we'll try and stay on time. Otherwise, she'll go on Wednesday. Great. We can see it. Go ahead.
All right, great. So my protein is CAM-KK2, and here at the bottom I have my composal script. All right, so CAM-KK2 is short for calcium calmodulin dependent protein kinase 2. That means that it is promoted by calcium and calmodulin. CAM-KK2 is a serine threonine kinase and phosphorylates multiple kinases, including CAM-K proteins like the ones listed on the slide, as well as AMPK. It is involved in the hippocampal activation of CREB1, as well as many other pathways, including but not limited to multiple MAP kinase pathways. And through the activation of the multiple CAM-K pathways, CAM-KK2 affects transcription factors, cell cycle, immune response, inflammation, memory consolidation, as well as neuronal growth. CAM-KK2 is found in many tissues throughout the body, but is predominantly found in the brain. It's found mostly in the nucleus and unstimulated cells and relocates to the cytoplasm and neurites after stimulation. According to Phoebus, CAM-KK2 is considered non-cytoplasmic, so I would assume that it spends most of its time in the nucleus and unstimulated cells. According to Phileas, it is a globular protein, and there's no coil domain. And while Phoebus predicted some transmembrane helices, these were located within my kinase domain, so this was likely a false positive. CAM-KK2 has one kinase domain, 4 low complexity domains, an autoinhibitory domain, and a calmodulin binding domain. And from the MODA output generated from my AlphaFold sequence, there does not appear to be any transmembrane helices or any other form of transmembrane structure within the protein, just small sites scattered throughout the structure that have scores indicative of membrane interaction. From the research I've done so far, there doesn't seem to be much known about how CAM-KK2 interacts with membranes, but based off what I could find, I would hypothesize that it is a soluble protein. As mentioned previously, CAM-KK2 activity is promoted by calcium and calmodulin binding. There do not seem to be any acetylations, myristylations, farnesylations, or geranyl-geranylations on this protein. However, QPTM did identify 6 methylation sites, all of which alter lysine residues. And from GPS lipid, I chose 3 likely sites of palmitoylation based on the amino acid sequence and the score from that table there. There are an abundance of phosphorylation sites in this protein. However, the important sites are likely the ones located in the autoinhibitory domain and the calmodulin binding domain. There's 1 serine which is phosphorylated in the autoinhibitory domain and 3 phosphorylation sites in the calmodulin binding domain. There's also 1 ubiquitination site within the calmodulin binding domain, and within the kinase domain, there were 9 phosphorylation sites and 11 ubiquitination sites. PIP stops and MET stops tend to inhibit binding to phosphoinositides, so these modifications likely inhibit membrane binding, and that would likely affect that large peak on my MODA curve around the 500 mark if it was interacting the membrane. For my disease-linked mutations, I found a quote from my paper which I think summed it up pretty good. So elevated CAM-KK2 activity is strongly associated with prostate and hepatic cancers, whereas reduced CAM-KK2 activity has been linked to schizophrenia and bipolar disease in humans. This makes sense as CAM-KK2 plays a role in multiple MAP kinase pathways, as mentioned earlier. The MAP kinase serves as pro-life signal, and so excessive pro-life signaling and not enough cell death is conducive to cancer. Its involvement in neuronal growth likely plays a role in the relation between downregulation and mental disorders. So mutations at positions 87, 139, and 268 result in decreased activity and would therefore be associated with schizophrenia and bipolar disorder. Mutations at 137 and 138 result in increased activity and are associated with prostate and hepatic cancers. And the mutation at 329 results in decreased activity. These mutations occur all throughout the protein and are not limited to one specific domain. These mutations occur in the kinase domain, low complexity domains, and outside of any formal domains, but potentially within a disordered region. This shows that mutations do not target one specific area or function of the protein, and that mutations in any part of CAM-KK2 could potentially lead to very serious health defects. And that's it. That's all I got. So I'll take your questions in the chat if you would like.
Great, Kelsey. Thanks. You did a great job with that and covered a lot of material as well, and very well presented. And Joshua, if you're ready to go, feel free to share your screen and You're welcome to start. And feel free to keep answering those questions in the chat space for the rest of you as well. Lots of good questions coming there. So do respond to your fellow students. We can see your screen, but if you could full screen it, that'd be even better. Perfect.
Is it full screen now?
Yeah.
Great. So the kinase that I'm going to be presenting today is CDK12, otherwise known as cyclin-dependent kinase 12. And as you can see, here's the composal script for it. It includes 2 polymethylation sites at residues 705 and 706. And phosphosite indicated there's many phosphorylation sites, but the main one I'm going to be focusing on is at residue 893. And it also is a homodimer. So as far as expression goes, CDK12 is a serine-threonine kinase that is expressed body-wide in all your cells. In the cell specifically, it's targeted to the nucleus, where it has its effect of regulating gene transcription. However, as can be seen by the predicted aligned error graph, it's very understudied, and the only domain which is well characterized is the kinase domain here. Everything else is very high error. As far as its domains go, it has 3 main domains. Kinase domain from residues 727 to 1020, indicated in red here. An RS, rich in arginine and serine domain at the N terminus, which has been hypothesized to help target it to the nucleus. And a C-terminal domain rich in alpha helices and a polyballistic cluster that binds ATP. For ligands, CDK12 is a member of the large family of cyclin-dependent kinases. So a large ligand is cyclin, specifically cyclin K for CDK12. Another important ligand is ATP, which uses transfer of phosphate to S2 and S5 position of the CTD repeats on the C terminus of RNA polymerase 2. As you can see, there's a lot of different CDKs. For regulation, CDK12 is regulated for the most part through cyclin binding. Typically, CDKs are around 40,000-fold less effective without their cyclins, so basically useless. Additionally, both CDK12 and CDK13, which is very closely related to CDK12, have a regulatory T loop at threonine 893 that's phosphorylated by the CDK activating kinase. And that's that phosphorylation site that I'm focusing on because it is the most important. As far as CDK12's pathway, it phosphorylates the C terminus of the large subunit of RNA polymerase 2, which then activates RNAP2 to initiate transcription elongation. And specifically, it's been shown to upregulate genes relating to DNA damage response and other cellular stressors. So it acts as a tumor suppressor, as you can see in the figure here. So hence, when CDK12 is mutated, it's been linked to many types of cancers, such as breast, uterine, and pancreatic cancers, and specifically HER2 and triple-negative breast cancer. So a lot of the papers I found on this indicate that it could be a very powerful therapeutic target for targeting different types of cancer, and specifically more aggressive cancers. CDK12 could be an important target for that. And there are my references, and I'm willing to take any questions in the chat.
Great timing and talk as well. Lots of information there again as well. And feel free to ask Joshua a question. We got a minute, perhaps before Grace starts our last talk for the day. Grace, that looks terrific. And maybe I'll just encourage you, Joshua, to answer questions in the chat space, and do ask Joshua and then Grace questions. If you can full screen that, Grace, you're good to go. Just unmute, and then you're good to talk your way through.
All right, so my kinase is the MAP/microtubule affinity regulating kinase 1. It is serine-threonine kinase, which is part of the calcium or calmodulin dependent kinase family and is considered to be AMPK related. It targets the motif KXGS, and which is found typically on tau and microtubule associated proteins 2 and 4. It's primarily expressed in the frontal cortex, though is widely distributed throughout the body, and it is commonly overexpressed in both Alzheimer's disease and autism spectrum disorder. Here we have screenshots of the disorder of the overall protein and the domains. Its catalytic domain is found from residues 60 to 311, with a ubiquitin associated domain quickly following from residues 325 to 370, and it doesn't actually appear to interact with ubiquitin and instead is more involved in the conformation and localization of the protein, as it is found within the cytosol, though can be peripherally membrane associated. The peripheral membrane association is mostly through the kinase associated domain, which binds phosphatidic acid, phosphatidylserine, and phosphatidylinositol 4,5-bisphosphate. This kinase associated domain also has the ability to auto-inhibit the catalytic domain. So here we have screenshots of MODA models with the red residues showing the predicted membrane binding sites. There are a few predicted membrane associations, though through most programs it was found that it's most highly expected to be associated from residues 699 to 710. And a couple really important phosphorylation sites involved in the regulation of the catalytic subunit are found on threonine 215, which is phosphorylated to activate it by liver kinase B1 and is expected to be inactivated or inhibited by glycogen synthase kinase 3 beta on serine 219. There are many membrane binding sites within, or many phosphorylation sites within the membrane binding region, which is expected to then inhibit its ability to localize to the membrane. One of the expected pathways that this is involved in is microtubule dynamics within Alzheimer's disease, because overexpression and increased activity of MARC1 can lead to the hyperphosphorylation of tau, and that inhibits its ability to stabilize microtubules and then contributes to neurodegeneration in Alzheimer's disease. Similarly, the overexpression of MARC1 can disrupt dendritic growth and impair neuronal development within autism spectrum disorder. And it's also expected to have it's known to have a positive action on the Wnt pathway, which in many cancers, but especially in colorectal cancer, is overactive and more expressed, which leads to the overall translocation of beta-catenin to the nucleus and enhances gene transcription and leads to uncontrolled cell growth and proliferation. And that is everything.
Okay, Grace, thanks so much. Thank you all for great talks. You've all put a lot of work into it. Feel free to ask Grace a question in person. If people need to go, I totally understand, but you can also ask questions of any speaker at this point. This is an open discussion. If you need to go, I realize you have other classes and other things on your schedule, but thank you so much all for presenting, and I will be sharing the video afterwards, and then I look forward to a similar session on Wednesday. I'm going to hang out here if anyone has a question or wants to chat. This is your space, your time for questions, and I look forward to seeing you next time. Thanks all of you for the amount of work you put into this. I learned a lot, so I really appreciate your time. Thanks for answering those questions, Grace. That's great. And I'm sorry, it's hard to go last, but you did a great job. All of you did a great job, so I really appreciate it. And I will share that chat as well, so you know what questions were asked in case you want to answer later. Okay.